cool cats and kittens.
Hello.
Hold on a minute.
Oh,
So sorry, there seems to be some minor technical issues where. Right, so let's transition that again. Okay, right now you look normal.
oh,
Large in
excellent.
life. So, hello everybody. I'm Faz Alam. On Twitter, I'm Defective Brain. In my spare time, I'm a science comedian, but my background is I'm a microbiologist, and I'm quite interested in running this microbiology journal club. And I'm here with my colleague Danny Chan.
Yep. Hi, I'm Danny. I'm also studied microbiology in graduate school, but these days I work in pharmaceutical advertising, fact-checking ads. I'm also involved in the DIY biology community, and I'm also very interested to dive into some of this primary literature surrounding uh, SARS-CoV-2
Yeah.
right now.
So
But I guess
yeah.
we don't. Maybe in the future we'll not talk about just this topic, but at the moment this seems like something very interesting to dive into, both personally, but also probably on a more global scale.
Yeah, we want to I mean, eventually we'll hopefully go back to normal microbiology, but we hope to eventually answer the question when we get out of our homes. But
Yes, yes, exactly.
so yeah, I mean, I didn't. So what I do now is I work for scientific journals. I used to work in research integrity, so I'm quite interested in the whole peer review process. And Microbiology Journal Club is an opportunity to see that peer review process in like real time.
hmm Yeah, and this is our off week, right?
Yeah.
On our on weeks, we try to run a journal club. We go through 5 questions that commonly would be seen in like an academic journal club. We chat out the methods and so forth. And on our off weeks, we're trying to identify the paper that we will talk about. So probably we'll like have a list and go through them, try to sense like maybe what's important. I don't know. We haven't really decided on any criteria.
Yeah, we're
um,
brand new. This is our official first episode. We won't talk about the lost episode, but
um,
yeah. So essentially, like catching up on the broader microbiology news, and of course, there is one news story in microbiology, which is COVID-19.
yes, absolutely. And so today and we encourage people to interact with us as well, right? Through
Yes.
on the on weeks Twitter,
Yep.
we used to do a Twitter journal club where people just chatted on Twitter.
So
So
yeah.
we still have that option. You can tweet us questions or suggestions for papers that you want us to cover.
Yep. And use the hashtag that's in the corner, which I'm pointing to right now. I'm trying to point to. Oop, oop, oop. Yeah. So yeah, I'm coping with the fact that screens are mirrored. YouTube's very new to me. I mean, I'm a lab person, man. If it's not in a pipette, I'm just
Yeah.
not understanding it. So yeah, I
Okay,
mean,
so we have
uh,
a big list of papers. Some of these, like we talked about when we were trying to choose the first paper we were going after. Um,
yeah.
but how do you want to go through this list? Do you want me
Well,
just
yeah. I mean, I'm thinking like there's like the main themes of the list we're going. So the things that we could talk about with coronavirus. So there's the treatments. There's like the nature of the virus itself, and like so I mean the main themes I was picking out was like the drugs, the vaccines,
Mhm.
the kind of population studies we can be looking at, and the antibody tests, convalescent plasma. These sorts of treatments are currently being tested at the moment. And I mean,
Mhm.
is there anything I'm missing as well? Like that I'm not
Yeah,
covering?
I mean, maybe like last week you talked about a therapeutic, but we also talked about the model, right? Like the
Yeah.
models that we're using to study this. We don't experiment in humans
Well,
for obvious. Well, usually, right? Like unless it's
yeah.
a clinical study. So a lot of the early observations we're seeing, especially when it comes to therapeutics, are coming from animal models, which can tell us a lot, but they also have their
Yeah.
limitations.
So I don't know if you heard about the news that the WHO have now like kind of approved human challenge testing. So so
Oh,
now
really?
yeah, they so this
That's
came
wild, actually.
I know. This like
Human challenge testing.
yeah.
So, actual challenge with coronavirus?
Yeah,
With
actually infecting people with coronavirus. They've approved that
what?
now. Yeah.
Human
So
challenge testing.
this came out like about a about a week or so ago. No, about 2 days ago. Sorry, they released
Two
the 2 days.
days
So
ago.
sorry, 6th of May. Sorry, so 4 days ago. Uh, and if you're watching this YouTube channel a week after the stream, then many more days ago. Time is relative on
Yeah.
YouTube. Um, So that's my excuse for not.
That's
Yeah.
that's wild.
So yeah, they've created some ethical like guidelines for doing human challenge studies. I so they released
Huh.
like this press release that essentially say that yeah, you're allowed to now infect humans with this, which is absolutely wild.
I did I did a search and I can't find the article. I see there's something from 2016.
Okay. I can forward you because I can forward you like a link to the article. Cause I got because I've been, I downloaded Zotero, which is the manager we use to manage our papers, and I've been playing around with it.
Hmm.
So I've got like a so I manage. Apparently, it works with the Guardian, so I can send you like a link to the study via the chat. Well,
Oh,
a link
absolutely.
to the news article about the study. I'm trying to find. So I've got the actual like kind of press release here that I'm going to forward it to you as well. Uh,
So I guess like because I found this report from 2016, it's like it's a topic that has been discussed before. It's not like they're rolling it out just for our current situation.
yeah,
But whether or not people actually have used it, I guess I'm not really sure.
I mean,
Human challenge.
yeah. So like for those,
It's specific.
yeah.
Oh no, I'm just saying it's specifically, at least the report I found, it says specifically for vaccine development.
Uh,
Human challenge trials for vaccine development.
yeah, I think so. Yeah, I think it is just for vaccine development. So let me. Oh, hold a minute. Oh, I don't have the.
Con- Conditionally vax trials. Interesting.
Yeah, it's all very. Can I send it to you via Skype? Maybe. Oh no.
I,
Yeah.
oh
Sorry. No. Sorry.
no, I got the link to the
All
page,
right.
and then I see that it's the Guardian page, and then it goes to this apps.who Iris Bitstream. We'll see what's coming down.
Yeah.
All right, 2019 ethics criteria. They've made many reports then on this, or at least two.
Yeah. So it's been kind of interesting for people
Hmm.
working in microbiology because this is the kind of thing that we've been tiptoeing around. That it's uh, so like the whole reason we use animal studies is because human studies have so many ethical issues that we need to deal with, and
Yeah.
WHO have just released some guidelines saying, oh, if you want to do some experiments on human, these are the kind of ethical
This
criteria
is
that we want you to look for.
that you'd have to follow.
Yeah,
Yeah. Hmm.
and it's
That's
yeah,
fascinating.
it's fascinating. I mean, I've been looking through it. It's pretty similar to what you do for animal experiments, basically. I mean, it's
Sure.
so yeah, but yeah, I haven't had the chance to really delve into deep. This is why I kind of this week I've been going on a tear looking at the vaccine studies that people are experimenting with. Even
Right.
so, yeah, I mean, there's something we could talk about, but
Hmm.
it's one of those topics that
Yeah. Doesn't really have primary, like, we're trying with this journal club, we were trying to talk about primary literature,
yeah,
right? So, I mean, fascinating. Thanks for sharing it. I'm like,
yeah,
it's cool for me to see. I'm not sure if we'll discuss it on the,
yeah, no.
yeah, more than just this,
In like
but
11 months'
so
time, we'll
fun.
probably be discussing it. Basically, this is like
Oh, perhaps. Yeah,
yeah,
that's true.
or 6 months if the things go wrong. I mean, who knows really? So I mean, it's
Hmm.
fascinating. I mean, it's one of those things that is. Oh, let me see. Oh, yeah, it's one of those things that's very interesting, and it might have lots of unintended consequences. Like you might think that oh
Yeah.
right, this will be help coronavirus research, but then also like there's all sorts of alternative medicine like therapies that also could be used for. So uh,
Yeah. Well, I mean, hopefully the considerations are like, right, probably a large part of it is like, it needs a good rationale to why to even engage in the study in the first place, right?
yeah,
Good previous evidence to say that this has some effect.
yeah,
Um,
and that's always difficult because who provides that rationale? How you define a good rationale? I mean, some people say, "Oh, you published lots of
Absolutely.
studies on this herbal remedy, and it seems to work in these guinea pigs." Or some, so yeah, I mean,
Hmm.
it's again one of those things that's quite hard to define, and it's quite interesting.
That's true. That's very true.
So yeah, I mean,
Okay. So let's dive into something. I
yeah,
mean,
let's hear some suggestions. I've got like my three vaccine studies that I want to suggest. So we could start off with those,
yep.
which are Uh, so, I mean, the first one is this study that came out. That's again quite hot off the presses. So,
Hmm.
DNA vaccines for COVID. So the rapid development. So this one
Yep.
is a preclinical
This is
study.
Smith
Yeah.
2020.
Smith 2020. It's actually preclinical, so they test it mostly on mice. But the interesting thing is that this.
Are there like, sorry, just one sec. Are there like new preprint servers that aren't like the archive from the archive family?
Yeah,
Because this is like Research Square. I've never heard of this one before.
yeah, everybody and their mum is starting a preprint server. I know the IEEE is starting a preprint server. There's like Eng Archive. There's all sorts of people trying to start their own preprint servers, and part of it is because like the UK, we have these rules that every like we I don't know, it's weird, so it gets very competitive. And oh dear, yeah.
Yeah,
So
it was my computer, I think.
okay, yeah. This for a minute there, I thought the stream was being destroyed again, and we had to rapidly move to
No,
make
we're
sure it
good.
stays okay. Yeah. So, and the interesting is like in Ovio, they actually been allowed to start bring their like vaccine. The people who are running this vaccine are being allowed to bring it to human trials, and also DNA vaccines themselves are something I find interesting because they're not just vaccines; they also are kind of gene therapies as well, which again is
Yeah,
interesting.
so the idea is that like the DNA is delivered and then it makes the protein, right, inside of
Yeah,
the patient.
exactly.
Yeah.
And once you like,
So it's
yeah, yeah,
yeah, like I mean like, you know, vaccines are interesting. There's so many ways to approach it and it's like when I remember talking about them in grad school, like there's a little bit of a black magic element to them as well. Like, you know, we have the basics of understanding that immunity happens
mm,
when you're exposed to foreign antigens, right? But the mode in which you're exposed to those antigens is super important for producing immunity. And people haven't really explored the full space of ways to expose people to those foreign antigens. Yeah, synthetic DNA. Do you know if there are previously approved synthetic DNA
I
vaccines?
don't think there are. I haven't found any, and it's. I mean,
Yeah,
I know that they were like,
because
yeah. Sorry.
here in the states they talk about this. Yeah, I said before we started the
Mm,
stream the Moderna vaccine,
yeah.
and that's synthetic RNA. It's
Oh,
the same idea,
yeah.
right? Just like put in the instructions, and then hopefully the body makes that foreign object and it gets recognized as foreign, and antibody responses mounted. There's a lot of people put a lot of weight in this new platform.
Mm,
You know, this is sort of like a turning point in the history of
yeah.
vaccine creation because it's very quick to make up a new synthetic DNA
Yeah.
vaccine, right? And if yeah, but I guess the safety concern still is that the protein is being made in the body somewhere. So each
Yeah.
one still has to be evaluated in its own way, but you can just it's a much more flexible way than having to make tons of protein. Making DNA is easier than making tons of protein.
Yeah. True. And yeah. So it's and the only different thing about DNA things is the actual like delivery of it, because I know that was a big bugbear for gene therapy, like trying to actually get the DNA into the place you want. I mean, I've read all sorts of
Sure.
like news stories about people using electroporation, which is basically like electrocuting cells until
In
they suck up the DNA, which is
in
in
humans?
humans. And I know like
That's so odd.
one of my professors was like did a trial on that for he was working on Duchenne muscular dystrophy, and it was completely
Huh.
impractical because it worked, but it was like only he had to inject this like square of skin, and it worked on there, but so
Yeah, that's kooky.
yeah,
Um,
it's kooky. But if they find a good delivery mechanism that works in human and is proven in a clinical trial, then that would be
yeah,
something
like
that's
I think the RNA one, it's like liposomes,
yeah,
right? They kind of,
yeah,
mm, I mean, which is, I mean, I love this. Like, our technology imitates life, right? Like, what is RNA or a DNA molecule wrapped in a liposome?
yeah.
That's a kind of like a virus.
Yeah, basically. I mean, it's and like liposomes. Yeah,
Except we designed it, right? Like
yeah,
we know the code, and it's not made to replicate. It's made to create this antigen.
yeah. I mean,
Hmm.
that's like so. For those who don't know, liposomes are basically little fat globules that can go
Yeah.
around your body and they can merge with your cell membrane and then dump their contents inside. Uh,
Yep.
and the great thing about them is that they're supposed to be very very safe. You can inject them and you're not going to get an immune response because they're made out of the same kind of fat that your own body's made out of.
Hmm.
So,
Hmm.
again, the big problem with like gene therapy that people are worried about is like massive uncontrolled immune responses that can cause severe like side effects. Um, but yeah,
So we have the DNA vaccine is one
yeah,
option.
and the other one is the only other trial that seems to be in humans. Which let me see if I can let me pull this up because there's a group that was based in China that came up with uh, yeah, an inactivated vaccine for COVID-19. Um, again, so this one is uh, so let me think. I mean, this was again. There's vaccine studies on COVID-19 are quite thin on the ground, so it's good that they had something. Let me check. Did they?
Hmm.
Because I think this one actually looked at. No, it doesn't. I think this is still a preclinical one. So again, this looks in non-human primates. So they got,
This is Gao et al.
yeah, Gao et al.
Rapid development
Yes.
inactivated vaccine. So this is a classic. These are the classic forms of
Yeah.
vaccines when you take a virus that was infectious and you treat it somehow so it no longer is infectious.
Yeah.
That's usually heat. Heat is a common way,
Mhm.
but formaldehyde can be used. That was one of
Yeah.
the original ones, right? Formalin
Oh yeah.
inactivated, just linking all the proteins together so they just
Yeah.
can't do anything anymore.
Yeah.
Basically, like a plastic molecule
Yeah.
that has the features of the real virus. So the right shapes that the antibodies made towards it would be effective.
Yeah. They're plastinating the molecules, so they're like
Plas-
like
yeah.
Gunther von Hagen's museum of viruses. So yeah, that was one that interested me. Another one that because I'm based in the UK, we've been going on quite a lot about this new big giant the study based in Oxford on Chadox, which is this brand new vaccine candidate that is based on chimpanzee adenoviruses, and
Mm.
they don't really have any data out for Chadox, but it was apparently given like approval based on previous results where there's a version of this like vaccine that instead of delivering coronavirus well, COVID antigens, it delivered MERS antigens. And
Yes,
so,
this preprints out
yeah.
MERS.
Oh, I think this one is actually published in the Lancet. Let me. Oh no, this is another preprint. Sorry, I have a bias towards.
I wonder,
Yeah,
is it published now? No,
I
it's
think it
not. This is
might be.
still.
I don't know. Because there were two articles about this. One was published in the Lancet, and one was a preprint. I think I chose the preprint. Uh,
Mhm.
But yeah, and that's because again, I guess selfishly, preprints are a lot less well baked, so there's a lot more to talk about usually in them. So,
Mhm.
because you,
Yeah, that's interesting. There is like I'm pretty sure one of the vaccine candidates in trials in China is an adenovirus based
yeah,
uh, object.
I mean, adenovirus.
I wonder if that.
Yeah,
Oh no, I'm just wondering if that one would have more like data for us to look at. Like that might have some more human data if they have
I
like
mean,
an
yeah, that might be.
early. Yeah.
Yeah, I mean, yeah, there's like a because adenoviruses have been historically because I know they've been used for gene therapy, like as kind of like DNA delivery systems. So
Yeah.
they've been. I know that they're quite good for that, and they're good at like
Are we
being
getting?
kind of tame.
Yeah. Are we getting our first ones now? I think the first ones are out now. The first gene therapies in
Yeah,
adenovirus vectors. That's the uh just Luxturna. It's like a retinal disease.
yeah,
Um, and they inject yeah, they inject adenovirus into your retina and then have it express the missing protein inside of these retinal cells that are lacking it, retinal
yeah.
pigment cells.
I mean, that's actually a good. I mean, the reason why the eye is such a good place for that is because eye is like an immune privileged area, so there is a lot
Yes.
less chance of like a really toxic immune response. So that's been quite good. I think that one came out of China as well. I'm not sure, but yeah. So those are my three like pitches for like vaccine papers. Um,
Okay. Right, I didn't tell you my pitches before we start. I have one. It's I think this one's being hit down somewhat in the media, saying that it's a bit scare tacticy. It's called Spike Mutation Pipeline Reveals the Emergence of a More Transmissible Form of SARS-CoV-2.
yeah,
Uh,
I think
have
that
you heard of
I
this?
have heard
Did you
of
hear
this.
about this
I
one
did
in the
hear about this,
news?
and there's been a lot of interest of it, and I get the impression that it might have been what the people say in the study and what the media have been saying about it. There's a disconnect, and it'd be quite interesting to
Yes.
delve into that.
Yes. Yeah, I think that it's just sent you the DOI. Uh, yeah, I think that this one is it. It seems like from what I was when I was browsing it, it seems like it's like a bioinformatics. You know, they did a lot of sequence gazing,
Yes.
and then they saw this, but they don't have any good experimental stuff to back it up. But I mean, it's kind of an interesting opportunity to talk about, you know, how does the in silico stuff and the observational stuff on genome scale translate into understanding something about, you know, real disease and what's actually happening in the population.
Yeah.
Because of course it's a very useful tool to be able to, you know, look at all these genomes and try to infer things, but you know it's not the be all end all, especially
Yeah.
as coming from the sort of experimental models side of it. There are things you just can't answer sometimes in a natural experiment. You need to have a controlled experiment.
Yeah, true. But
Yeah,
it is just interesting
experimentally controlled
to
experiment.
delve into those techniques and see like exactly because genetic drift is going to be inevitable in this virus. So seeing where it's going and where it's headed actually is quite interesting. And yeah,
Yeah. It could also maybe be a chance to talk about. I know that there's like a great website or something, and it maps out all the single nucleotide polymorphisms as the viruses spread. Um, and there haven't been a lot, and they're mostly non-coding, I think. But there's still this I think is for a coding mutation. Yeah, so just talking about mutations in general, right? And
yeah,
how that it would have yeah, we'd be able to talk about those types of things.
yeah, yeah. I
Um,
think that's a good thing to focus in
so
on. Yeah,
there is a section here about clinical outcome. Um, but I guess how are they doing it? I see. I guess they were also able to. I see, because they've matched the things that they sequence to samples that they isolate,
yeah.
right? So they can have a sense of where these people, how these people are going.
Yeah, so they look at the patients and they go back to on the data and go, oh, these people with this mutation, how did that turn out for those patients that we got that day from?
Right. Exactly.
And
Exactly.
yeah, then we can talk about yeah. So that's
Right.
an
But then there's
interesting.
all there's not that idea of like covariates, right? Like when you ask a question like that, you have no idea, right, how that was controlled.
Yeah,
Like there could
exactly.
be so many factors for why that disease went a certain way, but because we've lasered in done our science, right, we're like missing pieces of that.
Yeah,
Um, and then and then we have some papers that we talked about last week that I want to bring up again, like
yeah,
the profiling of, right, SARS-CoV-2 IgM responses
yes. That's
in convalescent therapy.
yeah. So convalescent therapy is another interesting topic because it's one of the oldest therapies, but also one of the most interesting ones to look
Yeah.
at. So
Oh, sorry, I misspoke. Not convalescent therapy, just like convalescent
oh,
people.
just looking
Just
so essentially to
yeah,
see if we like the state how we become immune to coronavirus.
yeah, exactly. Looking at what are those correlates of immunity potentially.
Hmm.
Oh, and then I had something about classic SARS that I wanted to talk about.
Ooh,
Just seeing like with the SARS, regular SARS, classic SARS. It's so easy to say.
yeah,
With
classic.
classic, yeah. There was a study done that said just to actually show the antibodies that did neutralize it.
Hmm.
Um, and there's lots of data there. Not a lot to rip apart, but a lot to
Hmm.
Um, I heard on This Week in Virology that SARS, the classic SARS, is classified as like at a very high as a select agent,
Hmm.
which is like a higher biosafety level.
Yeah.
Um, but SARS-CoV-2 hasn't been classified there yet. So it's actually at this point in time, you know, this is very based on the moment we are living in the moment, but at this point in time, it's easier to do work on SARS-CoV-2 than it is to do on classic SARS in the US.
I mean, the thing I can sort of understand because the case fatality rate for classic SARS was much higher than COVID-2. But the
Yeah,
problem,
yeah.
but the problem is the threat from COVID-2 isn't just the fact that it might it's the secondary infections, the infections you get afterwards. That's
Right.
the main issue. That so yeah, it's a tough one. But
Yeah, and then there's also a preprint that we talked about last time, potent neutralizing antibodies elicited by SARS-CoV-2 infection. So
mm,
that's a preprint again looking at
yeah,
a human population, I think. Is it
yeah.
human?
So
Let's
yeah,
see.
I mean, the interesting thing about the antibody, because something that I think was talked about with the original SARS outbreak was the fact that viruses can mess with the antibody response. So they can like the almost like distract it. So sometimes you get a high antibody response, but it's actually not neutralizing the virus, which I find
Hmm.
which could be an interesting aspect of these papers that they look at.
Yeah, and I mean, this is the sort of stuff that it's another aspect of the vaccine, right? Like vaccine is like getting these antibody responses, but part of what goes into understanding whether or not those antibody responses are good or not is seeing this side of it, right? Like
Yeah.
for people who are recovering, what does their antibody response look like?
Yeah.
Oh, right, and we had this. I mean, this one. This was also like a. I think it's also a preprint discovery of a 382 nucleotide deletion during the early evolution of SARS-CoV-2. So that's
Right.
not like as fear-mongering as the title about the other mutation, but there are. This is more about how it made the jump perhaps into humans, right? Because like if you look at the virus genomes that are infecting the bat population and so forth, I think that they don't all have this mutation. I think that's what this one's talking about.
Yeah. I mean, that's quite important. I mean, as I mean, it's, so where viruses tend to come out is like areas of high biodiversity, where in contact with human populations, and that's
Mm-hmm.
a big thing in
Yeah.
China, a big thing in like various places where viruses tend to come up. So, and a lot of people
And just
talk,
bats. I've
yeah,
I've
bats,
heard a lot of stuff about bats saying that like they have a very high metabolism for being flying mammals,
yeah,
and that necessitates also some flexibility in their immune system to withstand higher inflammation, I think is what it was said.
yeah.
Um,
I mean,
so they're also just like places that like the bats like I guess are particularly good at dealing with the immune effects of infection, right? Like
yeah,
they don't tend to overreact, I guess. And so, but you translate it into our context, like that's why that like the pathology of the pneumonia is immune mediated, right? Like we're using immune suppressants
yeah,
in some cases to try to get at ARDS, and so
yeah.
yeah, just
I mean,
that
yeah. I think there was a
how the
study
world is.
there was a study released like this week on like why bats have got like a super immune system, and like there's all
Mm.
sorts of things like I mean even flying animals, they higher metabolism means higher body temperature, which means that these viruses might be used to higher temperatures. The
Sure.
So our fevers
Yeah. So
do
they're
nothing.
do nothing. Yeah. Exactly. Exactly. Interesting.
What, so
Was that primary literature that you heard, or like
I
a bat
I believe
bat
so.
immune
So
system?
like so I mean it's basically I get these email notifications from Science Daily telling me of new studies, and one of them was
Hmm.
like, oh yeah, bats, and kind of it's something that we've been mostly aware of since like because bats have Ebola, bats was the source of MERS, there's the source of
Right.
they're thought to be the original source of SARS, and so like there are
Mm-hmm.
these spillover events from bats. Uh, so eventually someone will make the bats extinct, and then we'll be
Oh no.
just that seems to
That's like those how they try to get rid of mosquitoes because mosquitoes are like a big vector for viral transmission.
yeah. I mean, there was that big mosquito paper that came out about this bacteria that fights off malaria that I was contemplating bringing.
Oh yeah,
Yeah,
Wolbachia, Wolbachia.
yeah. So yeah, I was contemplating bringing that to here, but then I feel like there wouldn't. Our current theme is let's get out the house. No,
Yeah, that's true. We were trying to speak towards this
yeah.
pandemic more
So the next
directly.
malaria will get its own pandemic soon. Don't worry, we'll get to malaria.
Yeah, when the range of the mosquito increases a bit more.
Yeah, yeah, but I mean, it's like Zika virus, yellow fever, and all of them have like had like various pandemic versions, and malaria just waiting in the wings to do the same thing.
Mhm.
Because I mean, in Europe, malaria used to be a part of European life. Then we discovered DDT and killed all the mosquitoes, and
Oh gosh, and also any bird species that
yeah,
were particularly
I
susceptible
mean,
to
yeah, I
DDT.
think it was a sort of thing in the U.S. as well, and especially in the South. But I'm not sure. I need a citation
Oh,
from yeah.
no, definitely. I know that a lot of predator species,
Mm,
falcons, were impacted by the use of DDT at that time.
yeah,
Their eggs would become so thin that just nesting on them would crush the eggs. Um,
yeah,
yeah, but I mean, I mean, this is like, these are huge conversations about, the role of science and technology in fixing like huge global
yeah.
problems. Which is why, like, I mean, just circling back to that WHO news, right?
Yeah.
Like, which is so wild,
Yeah.
right? Because, like these decisions that we're making now in this climate, like they will stick with us in the way that we meet
Yeah.
challenges that are coming down the pipe. Yeah.
Yeah. I mean, and like it's super interesting because I feel that there will be like a kitchen sink approach in terms of vaccines because there's a lot of opportunity to like if it goes well and we have this DNA vaccine, suddenly a lot of gene therapy delivery systems will be like, oh, well, this is actually quite a safe. We can treat other diseases perhaps.
Yeah,
So
yeah.
potent.
Yeah, I mean it can be both like an incredible accelerant, right? Like bringing people like sort of uncovering new things that we maybe ignored because it was just easy to do things the old way,
Yeah.
right? It can do that, but then it can also like, from the regulatory point of view, like it can leave like a regulatory environment kind of in shambles a little bit,
Yeah.
right? Because like we've made all of these like provisional this, provisional that, right? And it's like now you got to rein it back in and like try to direct it towards,
Yeah.
something more.
And people are like, well, it was legal like 6 months ago. Yeah, but 6 months ago we're in the middle of a disaster. And yeah.
Yeah, I mean that's some news that I saw come from the FDA about antibody testing in the
Mm.
States, is that they never officially said that, all of these ones were approved, but like many companies went out and like made them. So now they're trying to rein it in a little bit and say like, no, like we originally said like only these labs could use it. So that still stands. Like we don't understand why this proliferation of products.
Yeah. I mean, the same with.
Oh yeah, and this reinfection. Sorry, I
Yeah, go
just
for it.
saw another. Right, it's reinfection could not occur in SARS-CoV-2 infected rhesus macaques. So we did talk about the rhesus macaque infection model. It seems like one of your vaccine things is also back in non-human
Yes.
primates. Um, but yeah, they also looked at whether or not reinfection could occur in these animals. Um,
Yeah.
so a place again where you can actually infect animals, see the antibody response that comes out,
Yeah.
make some comment about it.
Yeah. I mean, it's kind of interesting to see those studies now that people will be like, well, we can now literally do it in humans now. We can. So I mean, with Inovio, it looks like they went straight from mice to humans, which is just
Hmm.
super crazy. But it's interesting.
I mean, that happens. I mean, clinical one, the first, like if the safety profile is looking good in animals, I mean that's why clinical trial phase one
Hmm.
exists,
Yeah.
right? Is to figure out that dosing. Like in the first phase of clinical trials, people aren't getting doses that would have an effect on their disease, or like they're not likely to have an effect on their disease, because it's about finding the dose that is tolerated in people. That's like a really big concern in clinical phase one. So that's not too crazy to think that that they're jumping from mouse into humans right away. But I'm sure that the people who are making those decisions are feeling the pressure, right, to say yes.
Yeah.
Uh,
I mean, even for I mean, I did take a look at the hydroxychloroquine papers, and I felt so bad. Now I feel like we're bullying it with all the evidence that it doesn't work. I mean,
mm, mm, mm. Yeah, but I mean, at the same time, like it's great that that evidence has come out,
yeah,
right? Like we wouldn't have known if we didn't go down the trial path,
true.
right? Like, okay, ostensibly maybe people were too crazy. I actually read a paper recently, or I read an opinion
Mm.
saying that hydroxychloroquine is an established therapy in several other, right,
Yeah,
like things. So the increased demand for it is kind of ridiculous because like they need it to treat other
yeah,
things. And the paper took that point and then they extrapolated out and they looked at a table of all these different drugs that might have some effect in SARS-CoV-2, but then it compared them against the actual indications that they're approved for, right? And sort of saying that like, what does it mean? Like, healthcare systems have to take these things seriously because if you do take the kitchen sink approach, which isn't always even the right medical approach,
mm.
right? But if you do take that approach, it also has implications for supply for other
Yeah.
diseases.
I
Yeah.
mean, even if I know there's like a shortage of remdesivir because like I know the UK was going to have this massive trial on all these antivirals, and but
Mm,
remdesivir wasn't included because they couldn't get the stocks up, which is
mm, yeah.
yeah.
I mean, we talked about that last week. That drug isn't being used for anything right now.
Yeah.
So,
Well,
so
I did read recently that it's been used, that a version of it is used for animals, for feline a feline coronavirus. I believe there's a version of it out there,
ah,
but
I see.
yeah. So yeah,
So how are we gonna narrow this down? Like, what are we thinking is relevant to discuss?
so let me think. So
There are things that like might be around. Like last week, or not last week, but 2 weeks ago, we made some decisions saying that like, well, this might be around in 2 weeks, right? Like
yeah,
we didn't talk about the hydroxychloroquinone because we felt like everybody was talking about
yeah, yeah.
hydroxychloroquinone, right?
Everyone's beating up on
Um,
that drug. But yeah.
right, right.
No, you're right. So I think the vaccine will still be around next week. Like, I mean, there are,
And I mean, so will antibody tests. It's like
yeah,
not there's not so much that I feel is super hot there. We didn't intend to speak about such a topical thing last last week, right? But it's just that there was all this news that came out like a few days before saying that it had emergency use
yeah.
authorization.
So, I mean, the thing that be, I'd be quite interested in the DNA vaccine. I think you can probably tell because I've been talking about how it's game changing. It might,
That's true.
and then like the sneaky like news that the WHO has basically said, oh yeah, by the way, you can experiment on humans. You can give humans a disease. That's just
Hmm.
so. I mean, I feel like that is
Well, actually, that's well. They're not doing challenge trials though for the synthetic DNA vaccine, are they?
not that I know of. I mean, I don't know they're doing challenge trials for anything yet because
Anything right
yeah,
now, yeah.
because literally it was 4 days ago or that it was announced. So and since these guidelines are released, I imagine that people aren't working that fast to. I mean, maybe we'll see
Right.
some kind of trials
Yeah,
out soon.
like these trials are like probably this trial has been in the works for a while, right? It can't incorporate the new stuff
Yeah,
that was being said.
and even if you are going to infect people with coronavirus, you need to have a lot of preparation and stuff in order to get things right. Otherwise, you
Hmm.
like, oh yeah, we'll infect people, but we don't have any stores of coronavirus, or where do we get that from? How do we create a human challenge? What do you use to challenge humans with? Do we like,
I
yeah,
I almost want to study. I almost want to read the paper where they used it. Is Did they publish the paper where it's used in mice?
uh,
This synthetic DNA vaccine?
yes. So that's the one I'm linked to. So that's the one that they're using the DNA vaccine in mice. So I
Oh, the rapid development. But it's still a preprint.
it's still a preprint at the moment. Although technically, according to the website, it's been approved for Nature research, but uh,
I see.
so at the moment it's in production phases where so people are working to get the figures to look nice and
Yeah.
correcting the spelling errors. Uh,
Yeah. I mean, this is like part of the decision. Do we want published research or a preprint? And last week we chose a preprint, which was really cool because you had found that image that was different between that they had an issue about image duplication. That wasn't super
yeah,
significant, but just like an evidence of how messy preprints can be.
I mean
Um,
I've seen that happen in published articles. I mean,
right,
again,
right.
I work in publishing, so we reading our papers is like what we do.
Yeah.
Uh, So yeah, I mean, I worked in research integrity, which means that most of the papers I looked at had something wrong with them, even if they're published. So I've got a very cynical perspective on most published articles, anyway.
Which is, I mean, this is part of being a critical scientist out there, right? Is having that cynicism up, um, and just knowing the questions to ask, right?
Yeah,
That would bring you closer to understanding, like, whether or not something is acceptable. What are the standards of, right? What makes some data acceptable and others not?
yeah, uh, and that's super interesting. And it's quite. I find it quite useful to have these discussions out in the open, so that more people can like see. Because I feel like people get presented science facts like, oh, this is a fact, and don't see like how it's done or the messiness behind it.
Hmm.
And like rather than knee jerk going, oh, let's not trust anything, just go like, okay, well, let's develop our analytical brains, let's put on our skepticals
Skepticals, yes.
and see what we can and can't, because I mean, what we can and can't say from a study, and kind of what we can assume. Um,
Hmm.
so yeah, but yeah,
So very similar to the DNA vaccine is the Moderna vaccine, right? Which
yes,
is the RNA. And you said you couldn't find any papers on that one though?
I couldn't, but if you
That
can
was
find
the
something
issue.
on that, so I mean,
Hmm.
we,
I think it
uh,
would be cool to talk about a vaccine. That's like at least it also sets up to talk about immunology later or like try to talk about immunology later. We're not
yeah,
really
I mean,
immunology.
because I think I mean those antibody papers are quite interesting, and I feel like having something that later would be quite good. Uh, But yeah, I just like read this. I think it's the dealt two punch of the news that there's this vaccine coming out. There's this preprint that's just about to get like published.
Sure. Yeah, yeah. I mean, that's kind of nice. I see the immediacy of that one as well because, right, like this is its birth into the world,
Yeah,
its publication.
yeah.
Um,
And
and
if
we can
we
add our 2 cents or have people like ask questions about it as well.
exactly, yeah. And if we find some flaws, we'll be like, oh yeah, take that pharmaceutical industry. Ooh. Yeah. So yeah, I
Um,
mean,
hmm.
big.
Oh yeah, there are some papers that have been published for not specifically this virus, not novel coronavirus, but other viruses that are being used that use the same vaccine strategy.
Yeah. I mean, it is an interesting strategy that I'm because I remember like reading about these kinds of viruses that were used during the Ebola outbreak. Because again, when that came out, a lot of vaccine people saw an opportunity to try and test out. I mean, I feel like quite a lot of like half-baked drugs descended on Sierra Leone when the Ebola outbreak came out. But
Hmm. Hmm.
and that's always a
Hmm.
challenge. I mean, as a journal publisher, suddenly I'm seeing a big spike on papers that mention coronavirus or COVID-19, but don't really have anything to do with it. This
Yeah,
figure,
they're just
hey,
using the
yeah,
buzzwords.
because there's all this buzz of all the papers. If you have coronavirus, we're going to rapidly publish you. And the goes, oh yeah, brilliant. Well, if we can squeeze in like our
Right.
machine vision paper with a little bit of COVID, then suddenly we can get our algorithm published. And and it's kind of
Totally.
cynical how people. I mean, cynical how people are taking advantage of that, but not unexpected.
Right, right, right.
So yeah,
I see. Moderna has had clinical trials with Zika and influenza,
yeah.
but
So
not completed trials.
yeah, I mean, it's interesting because like a lot of these like kind of broad spectrum antivirals have been tested on multiple things before. So I know like there's one that I looked at called favipiravir, which I first encountered. I wrote an article on it like back in the Ebola outbreak because it was funny
Hmm.
because it's made by Kodak, and I'm like,
Kodak, the
yeah,
film company.
the film company, but a lot of those
They do a lot of
film
chemistry, I guess, right? They have like
exactly.
a chemical
Yeah, I think it was like
R&D lab.
oh yeah, it was either like Kodak or Fujifilm, one of the old companies that were made irrelevant once we got cameras on our phones.
Hmm.
But
Hmm.
so it's nice that they
But
found
they
like
but they
go on.
had to do research back then, right? Like they probably, yeah, they're
Yeah,
trying to retool their research departments to do things that maybe
yeah.
are related to health.
I
I
mean,
mean, human health is such a huge industry where we're talking
yeah,
about a slice of pie and of money in the world. Everyone, wants to be healthy.
yeah. Well, everybody wants to be healthy, but not me. I'm gonna eat my pizzas and not
Fast
exercise.
food.
Yeah, yeah, and unfortunately, our country's like coronavirus responses have been interesting to say the least. I mean, I'm from the
Right.
UK, you're from the US, and together
Hmm.
we're in a race to see who can botch the response best.
Yeah, well, we're moving into this mode of people want to reopen. Different states are deciding to reopen at this point.
Yeah, I know. I mean, yeah, I'm bored of this coronavirus. I'm bored. Let's just stop. Let's just go outside. It,
um, so wait, they in this paper, the rapid development, they reference something, reference eight. Did
yes,
you dive into some of these bibliography
I
things
did
that they
not get to
reference?
do that. I literally just looked at the title and looked at like kind of the
Because
abstract. And was like,
it's possible that we could find a paper in there that might have something as well. Although this has a lot of good figures. I just quickly go through the figures now. There's a lot
yeah,
of nice
yeah,
stuff to talk about.
yeah. It's
Looks
a
looks like a
lots.
very meaty
Yeah,
paper.
there's yeah lots of like tasty data to get your teeth into. So yeah, it looks quite good. Uh,
Ah, they use previously published was DNA vaccine induces SARS coronavirus neutralization. So classic SARS. They have a classic SARS.
the classic SARS paper. So yeah,
Published in Nature in 2004.
that's that this is a lot of faith. I mean, they've had 10 years to get this right, so fingers crossed. It should be okay. Um,
Yeah.
so yeah, I mean, that's what makes it
So that
interesting.
I mean that could be one that we look at as well, right? Like that's one. Because at the end of the day, they're both in mice, right?
Yeah,
Yeah, they're both in mice. As long as the question is too is like, you only have so much to talk about. Like you only have so much time to talk about something.
yeah.
Um,
So
and like trying to focus around like some big issues. Like in the last one, we tried to focus around the animal model, right? Like the
yeah,
monkey model um, and even preprints in some ways. Like uh, we tried to interpret the data. I think we also interpreted it. And with this one, there's a mouse model that's
yeah,
different as well, right? We haven't talked about the mouse model
yeah,
in either case. I've
yeah,
heard they're challenging models actually for the disease. Like
I
there was a
mean,
reason why some of these first papers were primate models.
yeah, I mean, mice, because I mean, we could delve into this a bit more, but compared to humans, mice have a lot of neutrophils. They have a lot of macrophage. They've got a lot. Their immune system is a bit stronger than humans, which means that a lot
Hmm.
of diseases that humans get infected by are actually very hard to give to mice.
Right,
So
right.
so you have to like give them like insanely high doses in order for them to show the same. And the way the mouse is like oral, like the way it's like respiratory tract is like set up is quite different for humans because mice don't
Mm.
have tonsils. They're obligate nose breathers, so they only breathe through the nose and eat through their mouth, which means that all
Hmm.
their immune stuff is up in their nose,
Right,
and
right.
it's kind of a subtle place to. So you're not going to see like a human response from them because there are some things that mice just won't be able to do that humans do. Mice don't get sore throats, but you can like try and see whether they got a similar like disease that can tell you something about the humans. And the same with so I mean, again, most same with like flu and other respiratory. So I
Yeah.
know for flu, the model of choice when I was working was in ferrets,
Ferrets,
or
yeah.
like ferrets or guinea pigs. And I
Mhm.
think like they use guinea pigs in this study as well, but I need to double check that. Um, but yeah, there are all sorts of little subtle differences. Mice are mostly used because they're cheap and they're a lot easier to take care of than a lot of other animals. So like
Yeah, they don't need that much stimulation, right?
yeah, like you don't need to play with them for 2 hours a day legally, which you do have to for like ferrets, cats, dogs, and a bunch of other animals. But with mice, because they're kind of prey animals, you have to like kind of leave them alone, or they'll get stressed out a lot. But you give them
Mm.
like special toys so they can hide away from other animals. Lots and lots of like environmental enrichment, so and nesting material, and it's just something I used to like focus in on a lot during my PhD. So you can tell I'm kind of very passionate about like you have to give them soft food when they eat. You have to do all sorts of stuff. Like
In
the
Smith 2020, they do look at guinea pigs. Yeah, they do some humoral responses in guinea pigs.
yeah, I think because guinea pigs have good B cells. I think for the immune system.
Uh-huh.
So I'm not sure why, because I think I might have slept through that lecture. But
What? Oh, there's 7 figures in this one, and in the published mouse study, there are 4 figures.
so
Um,
so I
and in this one, it's more focused on the humoral, like looking at like, oh, we gave them this immune factor, so then what type of antibodies are created? And
yeah.
then the one that was done in classic SARS that uses the same sort of vaccine one, they go into adoptive transfer to show
Ooh.
like when you remove the cells from the vaccinated mouse and you put them in mice that don't have it, like these are the cells that give the immunity.
Yeah. I mean I, I mean I think both are interesting. I mean what we I mean, I'm obviously biased in favor of the first paper, but I feel like if we have
Mhm.
time, we can discuss something of the other paper, or even
Yeah,
like. So I think what I'll do is
I like it. I really like figure one in this, like just as a jumping off point, like
yeah,
seeing the spike proteins in Pymol or whatever they outputted it in,
yes,
and the comparison between them. I think that like that's something that's an image that I'd like to show and talk about. So
yeah,
I'm sold. I'm sold on the Smith 2020.
yeah, excellent. I mean, yeah. So I mean, we can talk about that. So we've got our announcement. We're doing Smith 2020. I'll put the
Yeah,
link in the YouTube description. That what people YouTubers call the doobly doo, and
the doobly doo. They
yeah, it's a
call
thing.
YouTube the doobly doo.
Saying the word description can be very, very hard, and honestly, having your own slang for things makes you seem cool and with the in crowd, which is what I'm trying to do.
Oh, I understand. I understand what you're saying. Yeah, check out the doobly-doo for
Check out
links
the
to the
Yeah,
links
check
to
out
the
the doobly doo
paper.
for the links to the study. But yeah, it's something that I find quite exciting. I mean, all this seems quite exciting, and I quite like this paper. I feel like there's a lot to dig into, a lot of meat to it that we can
Yeah,
talk
yeah.
about.
I like that too.
Yeah, but yeah, I feel like yeah we so yeah, there's so much going on with this coronavirus. So trying to pick a paper to focus on. I mean, some weeks we might pick on something that might be a bit different. So I don't know. Um,
Yeah, well, you have to make some sort of like trade-off, right? Like it can't always be the most relevant thing. Yeah, I mean, reading papers takes focus.
yeah,
You really have to sit down. You have to look through these figures, and I mean, that's the journey that
yeah,
we want to take people on, right?
exactly.
Is going through the data,
I
what it
mean,
means to read data and
yeah.
interpret it for yourself.
I mean, literally, the last paper they made their data online on Figshare, and I downloaded all the data and reanalyzed it in my own in GraphPad to double check it because I get
Right,
paranoid
right.
about that sort of stuff.
Yeah, and I was looking at figures, not just the figures that were presented to me in front of me, but looking at the past figures that were published in different papers,
Yeah,
right? And how that data sort of like informs the design of the paper that we're seeing in front
yeah,
of us. Because science is also like right, it's in dialogue with all these other papers being published, and you want to see like yeah, they showed this, but was another group able to show a similar thing, or was another group able to show evidence that correlates or supports the conclusions that they're making in the current paper?
yeah,
It's a large task to read a paper, really. Yeah, you could
yeah,
spend a lot of time digging in, and again, it's about that level of detail that you choose to dig in with.
yeah.
Um,
I
yeah.
mean, I generally give myself about 3 hours for a paper that I'm excited about and 6 hours for a paper I'm not excited about. If I'm not excited about a paper, I really have to work hard and give extra
Ooh,
time because there's
that's a slog though. Yeah, 6 hours. But yeah, I can understand it.
yeah, not in one go
Getting all
maybe.
the
I'll just
pieces.
I won't chew through a paper in one go usually. Only if
Right.
but I mean if I'm really passionate about, it, I might just sit down and go, wait a minute, this seems wrong or this seems and then because usually I have printed out and I scroll over it and just like write all my little comments in the corners
Hmm.
and cross out all the sentences that have grammatical errors in the introduction, whether through that always are. We're
Oh
scientists,
yes.
not English
This is
graduates.
your editor lens going on, right?
Yeah, because yeah, I mean we're scientists, we're not English graduates, so I can't expect people to have things like, oh yeah, a paragraph should be a sentence. Let's have one sentence as just one giant paragraph.
But I mean, this is again the reason why we're doing this
Yeah,
at some level as well, is that it helps to read papers with others, because if we're all focusing on different areas, we bring something different to the table when we discuss it,
yeah,
right? Like, like I may read one paragraph very closely and follow the literature trail down, and Faz is going to look at another one, and that's just going to bring our understanding of the paper closer, right? Like,
yeah,
we talked about this when we were setting this up, but papers are this mode of communication where you don't have to do all the experiments yourself to understand what's going on.
yeah,
Like someone else has gone and done that work, right? Like the
right,
long hours,
yeah,
years, months of bench work, and then they've distilled it down into this, visual slash text format in order for us to understand the science from their
yeah.
perspective.
Yeah,
Yeah.
I mean, I guess people like spend ages looking at the Great Gatsby, thinking, what was Scott Fitzgerald thinking? Whereas scientific papers,
Hmm.
they want you to know what they're thinking. They literally, that's
Yes.
the point of the entire methods section, and that's why
Yes.
they're
Yeah.
kind of an interesting piece of literature for me to look at because they do
Yes.
want you to walk through their footsteps and see how they
Yeah.
saw the world. So that's
Yes.
why I
But we
find
don't. But we don't have to believe them, right? And that's the other thing.
exactly,
It's like they want you to, but as readers, we're also saying like, I'm not sure if I should.
exactly, and
Yeah.
that's why I like this idea for a for a journal club, and that's why I'm so glad that you're up for that as well, and that we're doing
Oh
this.
yeah,
Yeah,
absolutely. We're aligned on that matter, like why
yeah,
this is somewhat important to do, or why this is a worthwhile activity. And I'm enjoying it. I enjoy reading papers like this.
yeah.
Um, it's
Me too.
sometimes hard to,
I mean,
just by yourself.
yeah, the fun thing was when you get to like show you're working, because when you read a paper and you find something new, you've got no one to be smart at. You've got no one to say, look, I found something interesting. Look, recognize my
Yeah.
intelligence. Because when I'm doing that alone, I'm like, oh, okay,
Oh yeah,
no,
that's
nobody
right. I
cares.
mean, we are broadcasting on YouTube, right? There's a showboat element to this.
Yeah, there
A
is.
slight,
So I'm definitely trying to
slight
bring that.
showboat.
So now there's
Oh
an
man.
entirely new aspect to this, where in the corner I'll be writing jokes that I can perhaps bring up. Seeing at the comedy opportunities as a science comedian, trying to find the funny in difficult places because everyone's been on an airline now. We've all heard airline food jokes, but who's heard a PCR joke?
Yeah. Okay, that's good. I'm glad that we came to a decision. I guess we want to remind everybody that this is the format, right? On
Yeah,
one week we'll be trying to choose the paper. The next week we'll be discussing that paper. We want people to engage with us on Twitter. We don't have a big strategy right now to collect people. We're just figuring it out
yeah,
and learning as we go. But we have lots of energy and a background in infectious disease that we hope to share with people.
yeah. So thank you all very much for tuning in. Thank you for joining me, Danny.
Yep.
I guess this is where we say goodbye to the streamers, to the
Yes.
audience,
Yeah. Au revoir, YouTube.
or via YouTube.